Hello everyone. Good evening. Just confirm me that am I audible? Make sure that everything is perfect from my side as well as from yours. Everything is clear, right? Okay, good, fine. In today's class, I'm going to discuss MCQs from a topic cell cycle and cell division. But before that, I have to explain a lab operon concept. Okay, let's wait for 1 minute and let others also join and we'll continue the class. Okay. Okay. Okay, let them join. Okay, we have a lot to discuss today. See, generally, what is mean by operon? It is a cluster of genes. As I said before, it will be the cluster of genes. And where it is going to happen, it has happened only in the prokaryotic organisms, not in the eukaryotic organisms. Okay? Okay. Wait 1 minute. Let's wait for why nobody is joining. Okay. Generally, in lab operon, we are going to call them as the go on operon. Okay, go operon, or we can say it is always turned on when the inducer is present. What will be the inducer here? Lactose will be the inducer here. Okay, see, we have a different scenario here in the presence of inducer and in the absence of inducer. Okay, and before that, you have to know that this lac operon will be the go operon, which means what? Whenever the inducer is present or whenever the lactose is present, then only the lac operon will be turned on inside the body of the organisms, that is prokaryote, example bacteria. Okay. Okay, likewise, we have a promoter, we have an inducer, we have an operator, we have a lac Z gene and lac Y gene and the lac A gene. Okay, or we can, yes, lac A gene. And this will be the regulatory gene where we can, it promotes the RNA binding here. Okay, RNA will bind here, and where this will be the inducer site. In inducer, where we can find the this is the one going to induce. Whenever the inducer is present, they are going to initiate a reaction. And the promoter region will be here, where the RNA polymerase come and bind here. Okay, and we are finding the operator, where we are going to see the repressor. Okay. Here is the region where will be the repressor mRNA will be present. Through this, we are going to get the repressor protein. Okay. In this repressor protein, they are the one going to secrete the repressor, and this repressor is going to bind in the operator region. Okay. In the operator region. Now, whenever the repressor binds here, when we can find the no inducer, whenever the lactose is absent, the repressor go and bind with the operator. If the repressor binds with the operator, the RNA polymerase does not transcribe the operon, so that they can't see, we have a different genes and these genes code for a different enzymes. If the operator binds with the repressor, the operator does not allow the RNA polymerase to make the operon to on. Okay, this is what a condition when the inducer will be absent. Whenever the inducer is present, see here the repressor mRNA, this will be an inactive repressor. Okay, this will be an inactive. Whenever the repressor will be inactive, the operator binding site will be free, and the repressor will not bind to the operator, and this operator will be free, and it allows the RNA polymerase to continue towards the structural gene, and the lac mRNA will be produced here, and the lac Z gene is code for an enzyme called beta galactosidase, and the lac Y gene is code for the permease enzyme, and lac A is code for the transacetylase. Whenever the inducer is present, the repressor will be inactive and it is not going to bind with the operator, so that the operator will allow the RNA polymerase to continue to the operon, and all these lac mRNA is transcribed to the or translated to the protein. Are you clear with this? Or if you have any doubts, you can ask me now. Clear, everyone, correct? Just respond to my question, okay? Okay, see, let me ask you a simple question. Is it sufficient whether the repressor is not bind to the operator and the operator will allow the RNA polymerase and the mRNA will translate it to the protein? Is it sufficient for an efficient transcription and translation? Just answer for this question. Is it sufficient? Is there any other material required for the efficient transcription or translation? Hello? No, I'm asking, is that any material required for an efficient transcription? Do you know about the capside and cyclic AMP complex? Cap and cyclic AMP complex. See, this cap and the CAMP, cyclic AMP complex, they are going to attach to the cap site. Okay, cap site, and this cap site gives a positive signal to the RNA polymerase, particularly the C terminal. Okay, see, this cap and cyclic AMP molecule, it is going to attach with the cap site, and this is the site where when it is attached by this cap and cyclic AMP, it gives the positive signal to the C terminal of the RNA polymerase by the process of phosphorylation. Okay, then this RNA polymerase becomes active. Once it is active, it crosses the operator and it happens with an efficient transcription. If 100% efficient transcription has to occur, the first condition will be the inducer has to be present. Here the inducer will be the lactose. Okay, and the second will be the presence of cyclic AMP and the cap molecule. Okay, this is the two important thing which is required for an efficient transcription in the lac operon. Okay, everybody clear, right? Madhuri, you are the one asked the doubt for that. Okay, you clear? Okay, and I have a doubt why you people are not answering, you're not responding correctly. Okay, Anjaneyulu, why you are not responding? So many people are here, but nobody is responding. Okay, it is very important to respond whenever I'm taking the class, right? Okay. In today's class, see, I have attached what is the function of this promoter, operator, and these genes. Okay, lac I gene. It is a repressor. Okay, LAC-I is a gene. It is code for a repressor protein. And LAC-Z it is code for the beta-galactosidase enzyme. LAC-Y it is for the permease enzyme. A is for the transacetylase enzyme. Okay. Okay. In today's class, I'm going to explain a cell cycle and cell division. I'll give some of the introduction about cell cycle and cell division, and after that we'll go to the question session. Okay, last half an hour will be your question session. Okay, what are all the steps involved in the cell cycle? And first will be the interphase, a mitotic phase, and will be the G0 phase. Okay, in the interphase, this will be the longest phase. See, what are all the points I have been underlining, just make a note of it. Okay, the questions will come only from that area. See, in the interphase, this will be considered as the longest phase of the cycle. Why it is considered as the longest phase? Because it is further classified into or divided into 3 stages: gap 1 phase, synthesis phase, and the gap 2 phase. In the G1 phase, it is the preparatory for the preparatory phase for the S phase. Okay, in the S phase, they are going to replicate. The DNA replication is going to happen here. And for DNA replication, it is not a simple process, and all the preparatory stage for replicating the DNA in S phase, it has been prepared in the G1 phase itself. No audio and video from you. Okay, good, okay, fine. Do you have any doubts? You can ask me. Okay, if there is no audio, what are all the things you have missed? I don't know that. If you have any doubts, you can ask me now, right? And see, in the S phase, DNA replication will be taking place. At the end of this phase, what will be the end product? Each chromosome consisting of the 2 sister chromatids. Sister chromatids means what? This will be the chromosome. This will be the sister chromatids, and this will be the non-sister chromatids. We can call them like that. And in the G2 phase, the cells continue to grow and prepares for the cell division. It will be the preparatory phase for the mitotic phase. See, G1 phase will be the preparatory phase for the S phase, and G2 phase will be the preparatory phase for the mitotic phase. In the mitotic phase, generally where it will be going to happen, whether in the somatic cell or in the germ cell. Mitotic phase is happen in the somatic cell or in the germ cell. Somatic cell means body cell. Okay, germ cell means a reproductive cell. In which cell mitotic phase will be happening, or in the both cell? In which phase, in which cell mitotic phase will happen? In the somatic cell. Okay, in the body cell. No, it is not occurring. It is occurring in the somatic. Mitotic phase will occur in the both the cell, and in the meiotic phase will be happen only in the germ cell. Okay, this is what a difference between these two. And in the mitotic phase, as a result of this division, 2 daughter cells will be present, and the daughter cell will be looking similar to the parent cell. Okay, this is the cell. This is the one undergoing a mitotic cell. As a result, we are going to get a 2 daughter cell. How this daughter cell will look like? Exactly as a parent cell. This is what mitotic phase. In case of meiotic phase, what are all the cell? 4 haploid cell will be formed as a result of this meiotic phase. It is also called as the reduction division. And this meiotic phase will happen only in the germ cell, that is a reproductive cell. And all these cells are not similar. Okay, because crossing over will be happen there. Okay, this is a difference. And prophase, prometaphase, metaphase, anaphase, telophase. These are all the stages, and majorly we will say prophase, metaphase, anaphase, and telophase. 4 phase will be coming under the mitotic phase. In prophase, the chromosome will be condensed, and the nuclear envelope will be start to disappear, and the mitotic spindle will be started to appear. Okay, and prometaphase will be the stages between the prophase and the metaphase. And what has happened in the prometaphase? In the prometaphase, the spindle fibers attached to the centromeres. Okay, and the spindle fibers is formed by the microtubules, and microtubules is nothing but a cytoskeleton. Okay, and in this metaphase, here what is happening in the metaphase? Here the chromosome will be aligned to the metaphase plate. Okay, in equidistant from two spindle poles, and in anaphase they are moved towards the opposite poles. And in telophase, what are all the things disappear in the prophase, everything will be reappear in the telophase. And cytokinesis means a division of a cytoplasm. Okay, now G0 phase. G0 phase will be calling as the resting phase. We can call them as the resting phase. And in this phase, the cells are not actively dividing. It is not actively dividing, but the cells are active here. Okay, but the cells are metabolically active, but it is not dividing. This is what a difference, right? Now, see, some of these checkpoints are present when it comes to the cell division. We have a G1 checkpoint, G1 and ES checkpoint, and G2 checkpoint. G1 checkpoint, it is going to determine whether the cell attains a correct size or not, whether all the cells receive a correct nutrition or not, whether is there any damage in the DNA. Everything has been checked by this particular G1 checkpoint. And G1 or S checkpoint ensures this checkpoint will ensure whether the DNA is damaged or not. If there is no damage in that particular DNA, it has to be entered into the S phase, and in the S phase the DNA has to be replicated. Okay, and in the G2 checkpoint, whether the DNA damage or not, whether they are going to check whether the DNA is damaged or not, and the DNA is completely replicated or not before it enter into the mitotic phase. This is what checked by the G2 checkpoint. And spindle checkpoint, and it is also called as the metaphase checkpoint. They are going to determine whether there will be the spindle fibers is properly attached to the chromosomes or not. Okay, that has been checked by the spindle checkpoint. And DNA damage checkpoint, it is activated in response to a DNA damage. See, this DNA damage checkpoint is active only whenever the damage occurred in the DNA. Okay. Now, next stage will be the mitosis stage. There are some of the important points I have added here. Mostly it is diploid. Whenever the parent is undergoing a mitotic division, and we are going to get a 2 daughter nuclei, and this is in the diploid stage, 2n and 2n. Okay, this is what a diploid and somatic cells. Okay, and in plants this happens in both haploid and diploid cells, but not in the animals. And in the mitosis, what is the purpose of mitosis? The cell has to grow and cell has to repair, and the genetic continuity, everything will be take care by the mitosis process. And in the karyokinesis means what? Nuclear division. This nuclear division occurs in 4 different stages: prophase, metaphase, anaphase, and telophase. And always the cytokinesis happens only after the karyokinesis has been completed. Okay, and what has happened in the prophase? Let me explain little bit elaborately. Chromosome will condense, start to condense. Okay, heterochromatin, we can say like that. And 2 chromatids attached to the centromere, and the microtubules started to arise. Microtubules come, they are going to change their structure into the spindle form, spindle fibers. And see, what are all the organelles disappear in prophase means Golgi bodies, nucleolus, endoplasmic reticulum, nuclear membrane. All these are going to disappear in the prophase stage. And in the metaphase stage, the nuclear envelopes completely disintegrate. Okay, and 2 sister chromatids aligned at the equator, that is a metaphase plate. And the spindle fibers from the opposite, they are going to attach to the chromatid when it as it located in the opposite poles. And in the anaphase, they are going to split the 2 sister chromatids. This is what happened in the anaphase. In the anaphase, the sister chromatids become the daughter chromosomes. This can be asked in the question. And the telophase, what are all the things happen in the prophase? The chromosome has been condensed in prophase. But here, the chromosome will be decondensed in the telophase. And the 2 daughter nuclei will be formed, and the nucleolus, endoplasmic reticulum, Golgi complex, in the 2 different daughter nuclei, it will be reformed. And after that, the cytokinesis will be happen. The separation of cytoplasm will be happen in the cytokinesis process. And after that, meiotic division. Meiosis division, it is also called as the reduction division because it is going to reduce, because initially it was a diploid, it is reduced to a single N and single N, haploid. That is why it is called as a reduction division. Haploid gametes during a sexual reproduction, gametogenesis. This is what happened in the meiosis process. And what is the purpose of meiosis? Meiosis is responsible for maintaining conservation of the number of chromosomes. Okay. See, initially we have 46 chromosomes. Okay. And mother have a 46 chromosome and father have a 46 chromosomes. And half of the chromosomes to the daughter nuclei comes from the mother and half of the nuclei comes from the father, and ultimately the daughter will get a 46 nuclei. How this is happening, this 23 and 23, how this is happening by the process of meiotic division. Okay, that is why maintaining conservation of the number of chromosomes. Okay, and this meiosis is divided into meiotic 1 and meiotic 2 division. And the most important statement, okay, kindly make a note of it, DNA replication occurs only once in the S phase before the meiosis 1. What has happened in the meiosis 1 and what has happened in the meiosis 2? In meiosis 1, the homologous chromosome will be paired, and after that, the non-sister chromatids will be recombined, which means what? Crossing over will be happen in the meiotic 1. See, hope you all know the basic understanding. If not, if you have any doubts, kindly ask me. Okay. See, one chromosome from the mother and one chromosome from the father. As I said before, in the mitotic division, the parent cell, the daughter cell will look exactly as the parent. Okay, this is what occurs in the body cell. But in case of germ cell, we are not look exactly the same as like our parents. Why? Because crossing over is happening because of the meiotic cell division. Here, some of the chromosomes from this some of the portion of the genes from this chromosome and some of the portion of genes from this chromosome are going to cross over. That is why we are not looking exactly the same as like our parents. Okay, because recombination between the non-sister chromatids. See, this two will be the sister chromatids, and this one will be the non-sister chromatids. This portion is getting exchanged. A recombinance will be obtained. This is what happened in the meiosis 1 division. But in the meiosis 2 division, this is exactly the same as the mitotic division. Okay, mitotic division. And at the end of the meiosis 2, we are going to get 4 haploid daughter cells. Okay, and keep on stressing the chronological order. Okay, prophase, metaphase, anaphase, and telophase. Likewise, in meiosis 1, we have a prophase 1, we have a metaphase 1, anaphase 1, and telophase 1. In prophase 1, we have a further stages, and this order is very, very important. Leptotene, zygotene, pachytene, diplotin, and diakinesis. In leptotene, chromosomes start condensing. Okay, it has started to condense. And in zygotene, the most important point, a pairing of homologous chromosomes. Synapses is characterized by, this is called as the synapses, pairing of homologous chromosomes. This is what as synapses, and the synaptomial complex will be formed. As a result, the chromosome appears bivalent, or we can say tetrad. This is nothing but this. Okay. This is what a bivalent, or we can say the tetrad. And in the pachytene stage, crossing over will occur. See, pairing of homologous chromosome will be occurring in the zygotene stage. Crossing over will be happening in the pachytene stage between the non-sister chromatids. Okay, and it is an enzyme mediated process. What will be the name of the enzyme? Recombinase enzyme. As a result, we are going to get the recombinance. In the diplotin stage, the synaptomial complex will be dissolved and the homologous chromosomes will be separate. See, the diplotin stage will be exactly antagonistic to the zygotene stage, because in zygotene stage, we are getting the pairing of the homologous chromosome. In diplotin stage, we are separating the homologous chromosome, and in which stage the chiasmator formation will be happening in the diplotin stage. See, except at the crossovers during the chiasmata. And these are all not important here. And in diakinesis, the nucleolus disappears, chiasmata separate, a nuclear envelope, it is also degenerating. As a result, at the end of this prophase 1, everything will be disappear. And in metaphase 1, bivalent chromosome aligned at the equator. Okay, and in anaphase 1, they are moved to the opposite poles. And in telophase 1, everything will be reappear. This is followed by the cytokinesis. See, always karyokinesis is the one followed by the cytokinesis. Okay, now in meiotic 2 division, see, meiosis 1 is followed by a short-lived and transient cytokinesis. What is the meaning of this statement? This interkinesis will be the short span of time. It has happened in the very short period. And there is no DNA replication happened in these stages. And in meiosis 2, it is like a mitosis. As a result of this meiosis 2, each haploid cell will form 2 more haploid cells. Okay, so as a result of this meiosis 1 and meiosis 2, we are going to get 4 haploid cells. Okay, the same thing will be happen here. In prophase 2, everything will be disappear. In metaphase 2, everything will be aligned at the center. And in the anaphase 2, everything will be moved to the opposite poles. In telophase 2, everything will be reappear. As a result, we are going to get a tetrad. Okay, this is what an overview of cell cycle and cell division. Now your first question will be, what happens during a cytokinesis in animal cells? What happens during the cytokinesis in animal cells? What about cell plate and cleavage furrow? See, yes, that will be in the plant. See, in the cytokinesis, during cytokinesis in animals, generally cytokinesis will be the division of cytoplasm. Okay, and the cleavage furrow will be formed in case of animal cell, cleavage plate in case of a plant cell. Okay, now what is the role of cyclins and cyclin dependent kinases in the cell cycle? This is all related to the cell division, particularly the checkpoints. What is the role of cyclin molecule and a cyclin dependent kinases? Always the kinases related to what? It is adding the phosphate group. See, cyclins and CDK, cyclin dependent kinases, they are the one regulate the cycle by activating the various checkpoints. Okay, and the CDK is going to activate some of the checkpoints by adding the phosphate group and activating the checkpoint sometimes by removing the phosphate group also. It can going to do a phosphorylation and dephosphorylation, right? Which checkpoint ensures that DNA replication has been completed accurately before enter mitosis? Before it has to enter into mitosis, something has to check. And what will be the name of that checkpoint? Okay. Yeah. See, G1 phase. After that, what is going to happen? S phase. And after that, it is followed by mitotic phase. And after that, G2 phase. And after the G naught phase, we can say like that. Okay. So, DNA replication has been accurately before it has to enter into the C. Let me show you the picture. 1 minute. One minute. Okay. Sorry. G1 phase. G1 phase, S phase, G2 phase, M phase. As I said before, G1 phase will be the preparatory phase for the S phase, and G2 phase will be the preparatory phase for the mitotic phase. In mitotic phase, we have a further division. So answer for this question will be the G2 check. Okay. Now, what is the significance of crossing over during meiosis? Why the crossing over has to happen? It ensures a genetic variation among offsprings. It ensures accurate segregation of chromosomes. It prevents the formation of gametes. It promotes a mitotic cell division. Very good. And what if no crossing over happens? What if the condition, if the crossing over is not happens? What if the crossing over doesn't happen? Yeah, every human being will looking exactly the same. Okay, like our ancestor. This is what happened if crossing over not happens. It ensures a genetic variation among offspring. Okay. Now, what is the role of spindle fibers during mitosis? What is the role of spindle fibers? And this spindle fibers is formed by this spindle fibers is formed by spindle fibers, very good, microtubule, right? That will be nothing but a cytoskeleton. We know that what is the function of this spindle fibers is responsible for separating the sister chromatids. And which of the following is the characteristics of meiosis but not mitosis? Synapses and crossing over, formation of two identical daughter cells occurs in somatic cells, results in diploid daughter cells. What will be the characteristics of meiosis but not the mitosis? Okay. Synapses and crossing over. All others will be happened in the mitosis because two identical daughter cells will be seen in the mitosis. Body cells will undergo a mitosis process only, and diploid daughter cells will be produced as a result of mitotic division. Okay. Which enzyme is responsible for unwinding the DNA double helix during DNA replication? And in which phase is responsible for DNA replication? Which phase is responsible for DNA replication? Very good. Helicase will be the correct answer. And what about this topoisomerase? Very good. Yes phase. And what is the function of this topoisomerase? What is the function of topoisomerase? It cuts. Is it so? Remove. Remove what? See, whenever the helicase, see, DNA will be look like this, right? DNA will be look like this. Whenever the helicase starts to unwind the DNA, okay, it starts some of the tension in the DNA. Okay, it creates some of the tension in the DNA. It undergoes a supercoiling. Okay, and the supercoiling was removed by this topoisomerase. And what about this DNA polymerase? They are the one going to add the nucleotides. And DNA ligase is the one, if there is a, it is going to seal the nick. Okay. And the most active stage of cell cycle is, prophase, metaphase, telophase, and interphase. Exact. Interphase will be the most active stage of the cell cycle. Okay. Because interphase consists of what? They are consist of G1 phase, S phase, G1 phase, S phase, and G2 phase. Everything will be under the interphase. Okay. Which of the following does not occurs in anaphase 1 but occurs in anaphase 2? It is a meiotic 1 division and it is the meiotic 2 division. And that has happened in the anaphase, it does not happens in the anaphase 1 and it has happened in the anaphase 2. What will the process? Condensation of chromosomes, poleward movement of a chromosome, contraction of a spindle fiber, splitting of the centromere. Very good. Splitting of the centromere. Crossing over takes place in which stage? Crossing over occurs in which stage? Crossing over occurs in which stage? Just now I have explained you why. Packaging stage. Why you people are getting confusing? Packaging stage will be the stage where we can find the crossing over. In zygote stage, what is going to happen? It's okay, no need of sorry. Just aware of that, okay? Zygote stage, pairing of homologous chromosomes. Good. Which chromosome may be lost during the cell division? Which chromosome may be lost during the cell division? Is it telocentric? It will be an acentric chromosome. Acentric chromosomes mean what? A chromosome which is not where we can't find the centromere. Okay, that type of chromosomes will be lost during the cell division. Okay. Now, meiosis does not occur in ovule, anther, microsporangia, shoot tip. Because all other will be undergo the process of reproduction. They are somewhat contribute something to the process of reproduction, but not the shoot tip. Okay. Now, in which stage of mitosis the chromosomes are composed of two chromatids? In which stage of mitosis the chromosomes are composed of two chromatids? See, prophase and metaphase have the two chromatids. Okay. In anaphase 1, each chromosome is composed of. In anaphase 1, each chromosome is composed of how many chromatid? in anaphase 1, we are having a 2 chromatids. See, you are confusing with the number of chromatids at the different phases. So have a look at it, okay? Refer your book, refer sir's class, and make sure that how many chromatids are seen in the in each phase. Okay, this is what our homework, because you are lagging in this area. Okay, so this is your weak area, so you have to focus that. In meiosis, division of centromere occurs during. When will be the centromere will be divide. They are occurs in the anaphase 2. Very good. In meiosis, division of nucleus is twice, but the replication of DNA is only once. Division of nucleus is twice and the replication of DNA is also twice. Division of nucleus is once and replication of DNA is also once. Division of nucleus is once and the DNA replication is twice. They are going to give rise to a 4 haploid cell. Remember that and give the answer. This is the statement I have been underlined. DNA replication occurs only once. Okay. Where is it? Which questions? Okay, DNA replication is occurs only once and the division of nucleus will occur twice in meiosis. Okay, the correct sequence of prophase 1 meiosis is, you have to arrange the correct order. Liptotine, zygotine, pachytine, diplotine and diakinesis. Okay, if the cell is diploid in G1, then after the S phase, the cell remains. If the cell is diploid in G1, for example, we can say like this, okay? And after the S phase, the cell remains. Can anyone give the explanation? See, even after the S phase, the chromosome number will be same exactly in the G1 phase. Okay. Because they are in which form? They are in which form? They are in the form of chromatin. They are not converted into a chromosome. Okay. So it is always be a 2n. Okay. Don't get confused in this point. Chiasmata are formed as a result of. Chiasmata is formed as a result of exchange of parts of paired homologous chromosome, exchange of part of unpaired homologous chromosome, duplication of parts of parts of paired homologous chromosome, loss of parts of unpaired non-homologous chromosome. We didn't read this type of statement. In zygotine, what is happening? The homologous chromosome will getting paired. Okay, in zygote stage. In the packeting stage, the crossover will occur. The part will be exchange. Okay, and after that, in which stage? Diplotin stage, this chiasmata will form. Okay, this is what happens here. Synaptomial complex is a characteristics of mitotic chromosome, liptotic chromosome, paired meiotic chromosome, and metaphase. When will be the synaptomial complex will be formed? In which stage? In if you know which stage, you can easily answer this. It will be happen in the paired meiotic chromosomes. Okay, it is not in the liptotin stage. Crossing over that results in the genetic recombination in higher organisms occurs between. Crossing over occurs between a non-sister chromatids of a bivalent, two daughter nuclei, two different bivalent sister chromatids of a bivalent. We know that that will be happen in the non-sister chromatids of a bivalent. Okay, now arrange the sequence. Arrange the following events of meiosis in the correct sequence. Okay, terminalization of chiasmata, crossing over, synapses, disjunction of chromosomes, dissolution of synaptomial complex. What will be the correct order? Homologous chromosomes. Okay, only after that the crossover will occur. We know that it is in the zygote stage, it is in the packeting stage. Okay, and after the crossing over, the synaptomial complex start to form. Okay, only after that the chiasmata will be terminalized, and after that disjunction of a chromosome. So the answer will be option D, right? So the order plays a very important role. I think we have left some of the question here in the middle. In cell cycle, changes of which stage are not visible under microscope? These are all the simple questions. I want you to know these things. The question are less possibility to come in the exam, but make sure that you know these. Yes, interface will be the correct answer. Okay. How many chromosomes will be there in human cells produced by the meiosis? Meiosis produce how many chromosomes? Cells with how many chromosomes? Very good. Because it is going to produce an haploid. Okay. Diploid means 46. Haploid means 23. Okay. Division of nucleus is indirect. That is, nucleus is not visible during cell division in. These questions are not come in the exam. Don't worry. This will be childish questions like that. Mitosis. Okay. DNA content is doubled in the ES phase. Okay. That's all about today's MCQ series. Hope the session might helpful to you. And do you have any queries? Any doubts? Any suggestions? Any modifications to be done? And for tomorrow, what you're going to read? For tomorrow, we read cell replication, repair, and recombination. Okay, this is what tomorrow's topic. Okay. Or we can combine with the protein synthesis, DNA replication, translation. Just tell me, don't be quiet. I'm asking you a question. Okay. Tomorrow will be the replication, repair, and recombination. Okay, this is what your tomorrow's homework you have to read before enter into the class. Okay. Okay. If you don't have any doubts, you can leave the class. And if you have any doubts, please stay here. Yeah, sure, sure. I sent to the sir. He will upload. Okay. Yeah, everything, everything. Okay, 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 okay. All the class link, all the PPTs will be shared immediately after the class. And with respect to the class link, you have to ask sir. About the PPT, I'll say I'll upload in the group. Okay. Okay, shall we wind up? Yeah, okay. Thank you.